Welcome to Venture in the South. These are the stories of innovation and success in the southern U.S. that make money, have fun, and do good.
Welcome back to Venture in the South, a podcast about angel investing and early stage companies in the southeastern United States. I'm your host, Paul, this week, and I am here with Bill Stilley, the CEO of a company called Adovate. We're going to introduce you to Bill and what Adovate is working on and talk a little bit about some of the things that are happening in the world of adenosine. So, Bill, welcome to the show.
Thank you. Really appreciate you having me on.
Happy for you to be here. Would love to start this off with maybe you just giving us a quick high-level introduction to what you're doing and a bit about your background.
Yeah, first of all, again, thank you. So we are building a company called Adovate. We think we have all the pieces to have a major pharmaceutical company here in Central Virginia and in the Southeast. So that's what we're doing. And the way I got into this is, first of all, after I went to the University of Virginia, went into the Marine Corps, came out, went to the business school. And I'll note, when I went into business school, while I was at business school, I actually said at one point that I think I can do a lot of different industries, but the one I never want to go near is pharmaceuticals. They're just way too complicated, too much regulation, a lot of different reasons. I said, I don't want to do that. So out of business school, I started a company. It was a roll-up of industrial braking and clutching company. So putting together these companies service big industries like paper mills, et cetera. And I put that together. And actually, my first acquisition was the Klein Company out of Greenville, South Carolina. So that was the first deal I did out of business school. About a year later, I ended up selling that to a leveraged buyout group. So I was a really finance transaction-focused individual that wanted to stay out of pharma. So ended up starting a couple other companies, ran a public company for a little bit. And as I was coming off, I got convinced by the CEO of this company called Adenosine Therapeutics to join him as the CFO. So I didn't know the first thing about life sciences, but he convinced me that I should join him and be his CFO. So I did that. And by the way, that's almost 25 years ago. And I'll mention that company again was called Adenosine Therapeutics. And you're going to hear that word adenosine a few different times. So fast forward a few years later, I ended up becoming chief operating officer. I ended up kind of learning the research, in charge of research, in charge of manufacturing. I did a couple different deals and we sold that company, really nice exit, made a number of millionaires in Charlottesville, Virginia. And we all went off to do our own thing. And I'll say, by having a company like that, there's at least 10 other companies in Central Virginia now that have kind of sprung out of the personnel that were involved in that. And I did a couple of things, including worked for the public company that acquired us, was head of business development. So I was doing deals. And then I started a company, took it public. And while I was running that, I got a call from the chemist that was the head of chemistry at Adenosine Therapeutics. And he had been a direct report of mine. So I knew him, I knew his family. And the call went said, "Hey, Bill, just to let you know, Cheryl, who's his wife, has been in the hospital. She got sepsis and she almost didn't make it." I'm like, "Oh my gosh, Robert, that's terrible." And he says, "Yeah, but it's okay. She's on the mend now. She just got released from the hospital. Seems like she's out of the woods. But the reason I'm calling you at the moment is I sat with her for 6 weeks in the hospital when it was really touch and go. And I was, she was in bed and I was staring at the pill bottle, staring at the walls. And I had this epiphany on how I could make chemistry modifications to some of our previous molecules and other people's molecules to break the problem that has inhibited adenosine drugs from being developed." He told me about it. And I remember he said, "Bill, I don't think I can do this. I know I can do it." So I wrote him his first check. We put him in a chemistry lab. 6 months later, he was making the molecules he had said he could do. So I bought him with the public company I was running at the time. Fast forward a couple of years, we had strategically, I thought it made sense to actually split the company back up. So I stepped down. I went and got the scientific founder of Adenosine Therapeutics, had conversation. Actually, I wanted to have collaborations with him because he was still in academia. But along the way, we had one meeting. He came in and said, by the way, his name is Joel Linden. He came in and said, "Hey, Bill, I got a better idea. Why don't I leave academia, join you, as your partner, and I want to join you also financially, so I'll put $1 million alongside you, and let's get the company going." So we started Adovate, purchased that division from my previous company, and in June 30th, 2023, we now have the adenosine platform that we're developing today with Adovate.
Okay, got it.
So anyway, that's how I got where I am and how I got out of not having anything to do with pharma to being totally devoted to the industry, what we do.
There's a lot of fun things we could unpack there. I like the fact you were an entrepreneur through acquisition kind of PE guy 25 years before it was fashionable to do that. And I like the fact that you're accidentally in an industry now that you said you were going to not be in. But I particularly like the sitting down in a stressful situation, deep work kind of epiphany story there on where this kind of breakthrough came. It's fascinating to hear where people come up with their breakthrough ideas.
Yeah, no, appreciate it.
Well, maybe let's take one step back for people who don't know what adenosine is and what the kind of science is behind what you're doing. Maybe you could give us the layperson's quick intro to.
Yeah, happy to. So first of all, I wanted to say that there are things in the body called receptors. So receptors are all over the place. And if you think about that term, what it means is it's basically a keyhole in a cell or an enzyme or a tissue where a molecule that's in the body will come along, be a key that fits into the keyhole, turns, and makes something happen within that cell or enzyme, whatever it may be. And so you may have heard of molecules that are made to do that, like serotonin, dopamine. So serotonin, for instance, is a key that gets released by the body at certain points, and it acts on 7 different receptors. So it's like a skeleton key that will activate or turn 7 different receptors that will make different things happen in the body in different places. And so dopamine acts on 4 different receptors, and you can have the naturally occurring activators like they exist in the body, or you can find in nature, or you can make them in a chemistry lab. But then you can also have the opposite, which is a blocker. So a blocker is like a key made to fit into that keyhole, but then it doesn't turn. It just sits there so that when the naturally occurring activator comes, it's not able to get into the keyhole and therefore not able to cause its action. So some of the antidepressants, they'll stop serotonin from being uptake, which means going away. And then you have things like a beta blocker, which would be a drug that blocks the beta receptor so it can't be activated. So adenosine is a primordial molecule of life. It's one of the absolute building blocks, at least in mammalian life, and it works on 4 receptors. And by the way, adenine, which is a piece of adenosine, is one of the 4 DNA building blocks. And throughout the body, there's a ton of different things that adenosine does through these different receptors. And you may have heard of a molecule called ATP, which stands for adenosine triphosphate. It's often called the battery of the cell. So what happens is the body will take adenosine, it'll put phosphates onto it, which is a type of chemistry group, which takes energy, store it in the cell, and when the body needs energy, it will take the ATP, release the phosphates so that it lets the energy go, and you get ADP, adenosine diphosphate, AMP, adenosine monophosphate, and then adenosine. And that's how the energy, and that's why the energy is released. And when that happens, you then have adenosine around, which will then activate the receptor. So ATP does some things, and then adenosine goes to work. And so some well-known effects of it are in the brain. The 1 receptor has effects that put you in the mood to go to sleep. On the heart, it's one of the things that helps keep your heart rate under control. It has a lot of bowel effects as far as helping your bowel motility. It helps creation of blood vessels. It's involved in the pain signaling and just 30 different things throughout the body. So now I'm going to ask, and you're ready to queued up on the video. I'm going to ask Paul to play a video that just shows in the brain. Remember, I said there's 1 receptor in the brain that works having to do with sleep induction, and just gives you an idea of how a receptor works in a molecule you may have heard of that works through adenosine. Go ahead, Paul.
All right, let's give this a try.
Your body needs a constant supply of energy, which it gets by breaking down a high-energy molecule called ATP. In the process, it liberates adenosine, ATP's chemical backbone. Neurons in your brain have receptors perfectly tailored to this molecule. When adenosine docks to these receptors, it activates a cascade of biochemical reactions that cause neurons to fire more sluggishly and slow the release of important brain signaling molecules. In other words, you get sleepy. Caffeine is what's called an adenosine receptor antagonist. That means it derails this process of slowing your neurons down by blocking adenosine receptors. Caffeine and adenosine have a similar molecular structure, close enough that caffeine can wedge into the adenosine receptors, but not close enough to activate them. To summarize, adenosine inhibits your neurons. Caffeine inhibits the inhibitor, so it stimulates you.
There you go. So,
All right.
so what you saw there is the one receptor that has to do with adenosine buildup that puts you in the mood to go to sleep. And caffeine actually works through blockade of the 4 adenosine receptors. So caffeine is what's called a non-selective adenosine blocker, which means it blocks all 4. So in the brain, stops you from getting in the mood to go to sleep. On the heart, it inhibits adenosine from keeping your heart rate under control, and it has bowel effects through that A1 receptor. So if you drink a lot of coffee, you can't sleep, your heart rate goes up, and eventually you have some bowel activities that related to drinking so much coffee. And so that's just one, and that primarily, that works on all of them. And in the literature, coffee has been used since the Middle Ages to treat asthma. It's been known until about 20 years ago, it was the coffee, 2 cups of coffee was the best treatment for Parkinson's. And caffeine is actually approved for one thing, and that thing is pain. Adenosine was invented in 1916. Excedrin's been around for 50 plus years. And the problem with using caffeine, though, is if you gave enough of it or high enough, you were able to concentrate and give enough of it that you could have a more material effect, for instance, on pain, you would actually explode somebody's heart because you would inhibit the ability to keep the heart rate under control. And so that shows how the non-selectivity can be a problem, but the adenosine receptors work across a number of things. So what we do at Adovate and what Dr. Thompson invented was a way to stay away from that A1 receptor and actually any of the receptors he wanted. So you only had activity on the receptor that you wanted to have an effect, and keep drug-like characteristics. So historically, you could either have effect on that A1 receptor, the one that has the cardiac effects, which by the way, that's the big problem, is the cardiac effects. You could either have effect on the A1 receptor and have a great drug as far as being able to get distribution in the body and get where you need to go, or you could have a drug that was selective. And nobody had been able to invent a drug that could get distribution in the body and stay away from that A1 receptor. That was the breakthrough. So it's a really simple idea. And I like to say at Adovate, despite the fact that we have a pipeline with 10 different drugs going on targeting major diseases, is that we're just a simple company. We go after proven pharmacology. So this is the effects of adenosine are well known, what they do. We're going after proven and known things just with a tool that can do it better. And with that, it gives us the ability to like I said, pain, asthma, cancer, diabetes, number of inflammatory disorders, Parkinson's, etc. All those things that adenosine is just well known to be involved in doing. And now we have the drugs that can actually target them.
Okay, got it. So you are more complicated than a regular drug development company in that what you can produce is really a platform then of different drugs using the same technology to target a whole bunch of different challenges, right?
Correct.
Yeah. But on the other hand, that does lead to some challenges about figuring out which ones to prioritize and how to raise enough money to develop all these different things. So what is sort of the main focus of the development efforts from here?
Yeah, absolutely. And that, and I would say any platform you have, whether it's technology, whatever it may be, something, let's say Kevlar, for instance, anything you have that has multiple potential applications, the first thing you have to do is figure out what's your killer app. If you try to do 100 things at once, you will fail at all 100. And so in our situation, what we believe our killer app is asthma. And so we have a drug right now that's in human testing, and I'll give a little, we have some breaking news, and this is, I know this won't be particularly secret, and we'll be announcing soon, but yesterday we dosed our last group in our phase 1 study. It was 1.5 grams, which is a massive dose. So we had a really high dose, and we had no drug-related AEs. Was reported to me about 10 minutes before I started this call. So what that means is we have an adenosine, our opinion, the first adenosine 2B antagonist. So there's the A1 receptor I've mentioned before. There's the 2A receptor, the 2B receptor, and the 3 receptor. By the way, they used to think there were just 3, the 1, the 2, and the 3. Then they realized that the 2 actually had 2 subsets. So now you have the 2A and the 2B. So the first adenosine 2B antagonist that has ever been able to be dosed orally, because our drug is expected to be an oral treatment for asthma, that has now been shown to have good levels in the body when taken orally. So that, in my opinion, gives us a drug. There's a few different things you could do with that, but we are targeting asthma, and all of our focus is there. So the other things we're working on, we're winning grants, and we're doing it with academic collaborators that are using their labs to do the work. So 95% effort on asthma, 5% to manage the grant program and the other academic collaborators. So once we've had success in the first one, we can then pick up the one that has the best data at that point in time. And I actually have a term for this. I call it the focused portfolio approach.
Okay, got it. And so the focus of your portfolio is asthma. So maybe just tell us a little bit about sort of what subset of asthmatics do you provide a treatment for that they don't have a treatment for already, and why this is different and filling a need for them.
Yeah, first of all, I'll say just by the fact you asked the question that way tells me you know something about asthma and about drug development right off the bat, because you're talking about the subsets. And let me say this. There is a drug on the market today, and remember I said we're going after known pharmacology with better tools. There's a drug on the market today called theophylline. Theophylline's been around for decades. Theophylline is the only, so it's approved, but it's the only drug that works in all types of asthma. So the only one. And the way it works is it is a non-selective adenosine blocker. So think about caffeine times 3 to 4. That's what you have with theophylline. So people that are on it, as you would expect, it works for their asthma because it's hitting that A2B receptor, but they can't sleep. Their heart's doing weird things. They have bowel issues. And in fact, you have to monitor blood levels. And if you get it wrong, you can die. So the FDA has given theophylline what's called an NTI designation, which is narrow therapeutic index drug. So NTI, which means that the difference between the effect and really bad things like death, for instance, is small. And so that's a step beyond what they call a black box warning, which is the normal warning. So theophylline is a dangerous drug. It's very difficult to tolerate if you're on it, but a number of asthma patients need it to live. And so the FDA leaves it on the market. So we believe that our drug will basically be a strong caffeine, because we'll be able to dose it higher because it doesn't have the cardiovascular effects, without the side effects. So even if we were just like theophylline without the side effects, we think we have a blockbuster that can go after all asthmatics. Because we can dose it higher, it might even be more effective. We'll see as we continue into trials. Now, with that said, our initial indication may be a subset, because that might be the easiest way to get to an approval. So for instance, we're looking at severe asthma as a really unmet medical need, and we might end up targeting that. But I'd say that decision will be made in the future based on the data we see in the initial human studies that we're doing.
Okay, got it. And I guess that's why it's a big deal there were no adverse drug effects with the high dose at the beginning here, is because if you had high dosed somebody with the existing drug on the market, that would have immediately shown you some negative effects. So you are proving immediately that it is selective and only targets the 2B receptor.
Correct.
Yeah.
And by the way, in the binding assays, which you run in Petri dishes, it's 10,000-fold selective over those other receptors. So effectively, it has no effect.
Okay, got it. All right.
Yeah, and I'll say, if I just a tiny bit about asthma, a lot of people don't realize how big a problem asthma is, and it's because there are no solutions, and so you really don't have much to talk about. But 400,000 people every year die from asthma. In the U.S., it's a few thousand, but globally, it's 400,000 people. So this is a truly unmet medical need, and the primary, what they call the first-line therapy for asthma are steroids. So if you're a serious asthmatic, you're on chronic steroids, which have a ton of side effects. That's really not a good long-term solution. It's the best we have, so I don't want to tell anybody to go off their steroids. The opposite of not having it can be worse than having it. But bone density, skin loss, psychiatric effects, just all the things that keep coming out worse and worse about long-term chronic steroid use. So we want to go and help those patients. And I'll say the last thing about this in theophylline, I mentioned theophylline is the only drug that works across all types of asthma, and it's because of how it works. So if you think about an asthmatic attack, there are really 3 components to it. First, there's the trigger. Something will start the asthmatic attack. It might be a pollen, something to that effect. So a lot of asthmatics, what's happened is their body has recognized a protein that is not a pathogen like a virus or a bacterium. They've created an antibody on what's called a mast cell. They've created an antibody, which is by the way, that's a receptor. An antibody is a type of receptor, which will then recognize, instead of the protein of a virus, it'll recognize the protein of a pollen or cat hair, something to that effect. And that will send a signal, that receptor sends a signal to the mast cell to go into an inflammatory response. So the second piece is the inflammatory response at the cell level. And that inflammatory response is called mast cell degranulation. So the mast cell basically spits out the cytokines, which are these cells that go out and create, they're attack cells. They go out to go and attack. It also puts out a ton of ATP, adenosine triphosphate. The adenosine triphosphate comes out, it releases its phosphates to put energy into the system, and then the remaining adenosine goes out and activates the A2B receptor, the adenosine 2B receptor, on the surrounding mast cells to bring them into the inflammatory response, tell them to also degranulate. So, maybe 1 in 10 billion mast cells might have that antibody, but once it triggers, it then recruits the surrounding mast cells, creating what they call a cytokine storm. So if you think about that process, all the other drugs out there either stop the trigger, they stop the inflammatory response somewhat or inhibit it. For instance, Singulair is a leukotriene inhibitor. That's one of the cytokines. So if you have Singulair on board, you will reduce the effect of that cytokine. Therefore, you will reduce the inflammatory response, but you won't stop it. And then you have the recruitment of the rest of the cells. So I like to say that the trigger is like, you're in a forest, you take a match, you light the first tree in the forest on fire. The flames are the cytokines burning that tree, but the adenosine is the wind that blows the fire through the rest of the forest. So we don't stop the match, and I'll say theophylline and what we expect us will do. We don't stop the match, we don't stop the flames, we stop the wind. Mm-hmm. And that's why no matter what kind of trigger you get, no matter what kind of response you're triggered to do, the promulgation is what's inhibited. And that's why theophylline works in all types of asthma and all the other drugs that are out there. So we would expect our drug to work in combination with the other drugs and, potentially in an additive effect.
Okay, got it. I am beginning to understand now why that is the focus of the focus portfolio. It feels like if you can develop that drug and get it approved and get it marketed, it's a mega blockbuster of a drug.
Yes, and in the end, you say we're actually a me too. We're not inventing anything new. We're just doing it with way better compound, and it'd be meaningful to millions of people a year.
Okay, got it. And so that's the focus of 95% of the company from here, is to go through the rest of the drug trial processes and get it approved, or is there a different sort of business plan here?
Yeah, so there is absolutely a different plan, which is and this is another reason why we chose asthma. So there is a study that's been done since the 1980s. It's a diagnostic test where you take adenosine, you give it to an asthmatic, have them inhale it, and they will go into bronchospasm. You say, well, why are you doing that? Why would you want to send somebody to bronchospasm? Well, what they do is they start at a really low, and by the way, healthy people will not respond. They haven't been primed this way. You give a really low concentration, and over 30 minutes, you step it up until the person gets to a 20% increase in airway resistance. So they just start to wheeze a little bit, and then you stop the study. So nobody ever gets into danger or anything like that. But what it does is two things. One, it confirms asthma rather than something that maybe looks like asthma. And two, it puts them on a severity spectrum. So if it took a little adenosine to get them to the 20%, then they're severe asthmatic. If it took a lot of adenosine, they're a mild asthmatic. So what we're going to do for our next study after this safety testing, which by the way, we expect to start in the next couple of months and be done this year, is we are going to do that study on and off drug. And if we can increase the amount of adenosine needed to get the patients to their 20%, then we would expect our drug to work if used as an asthma patient. With that data in hand, assuming success, we believe we are in the perfect position to sell that product to a major pharmaceutical, pulmonary company, which most of the major pharmaceutical companies are. So we would that We would sell then, we expect in 2026, that product. Then we'll have the proceeds we would distribute to our investors. I will raise additional money. My expectation is some of those investors will participate by leaving their funds in from that. And we would then develop the next 1, 2, or 3 out of the pipeline based on the data we have at that time. Again, looking at the efficacy, the need, and what's the, I'd say one of my big experiences over time is it's not just about the efficacy, the need, it's about what is it going to take to prove to the FDA that you can get to an approval. And so with asthma, we feel like we really have quick human proof of concept. We can do it in a 30-minute study given a couple of times versus, some 6-month study if you're trying to do, let's say, lupus. By the way, I love lupus as an application we're going to get to eventually. It's just not the one that gives me that first human proof of concept quickly. So we are going to do a lupus drug. I mean, mark my words on that, but just not first. It's not the first for it. And we have a couple of others where we think we can get to a really quick human proof of concept also. And eventually we will my plan is build this company. Most likely we'll get acquired along some way, but we're going to continue to build it, sell the first one, partner a few others, and then eventually keep some ourselves and take it to market.
Okay, got it. There's, I guess, 2 big objections that inexperienced or angel investors like me have when we're looking at life science companies. One is the bimodal outcome on the drug not working or not, and you solve that by having a whole portfolio of potential applications. And then the second is how long it takes to get to a point where somebody acquires the drug that's being developed. And your solution to that then is to sell them early or relatively early, having got some compelling data that they are going to work.
Yeah, correct. And I would say, depending on the drug, we might keep it longer or shorter than this one. This one we chose first because we could see how we get to human proof of concept, how we could have it positioned really well as an acquisition of the product, and move that one out the door. So this one was designed with the intent, the goal going into it was get it to a point that we can sell it. That will validate the pipeline, allow us to bring in funds and then.
Okay, got it. And I think another piece of hot off the press information is that you did raise some money recently. So can you tell us a little bit about what, from whom, and what the sort of use of the funds is going to be for that?
Yeah. So first of all, we announced that we raised a little over $6 million on Monday. We put out the announcement. We had actually targeted $6 million. Had some good news where actually the cost of our trials ended up being 1/6 of the cost we had expected due to some we put forward a more innovative design to the FDA, and they agreed. And so because of that, we actually dropped it to $3 million, and then we ended up a month and a half later closing on $6 million. So that's just how that went down. History of the company is when I started it, Joel and I were the initial funders. We raised about $7 million, get it moving forward, and take it to the point we are now. We've now raised another $6 million. That will allow us to complete that human proof of concept study and if I had to, I could make that money last through the end of 2026. We think that's probably not how we'll spend it. We'll probably spend it a little earlier because there are some other opportunities.
Okay, got it. Well, congratulations on raising a material amount of money in early stage life sciences in the Southeast. That's not something too many people are able to do. So congratulations on doing that.
Yeah, thank you. And I'll mention, I appreciate having Venture South and some of their team on board as part of that round.
We're happy to be on the cap table. Yeah. Maybe you could just tell us a little bit more about the sort of the challenges or the opportunities of building a life sciences company in Charlottesville, Virginia, which is, without trying to disparage anywhere, not known, I think, internationally maybe as a life sciences hotspot. Tell us about what it's like to build a company in this space in that kind of geography.
Yeah, so appreciate that. And there are absolutely some challenges. There's also some strengths. Lifestyle here, when you get people into the area, they don't tend to leave. As I mentioned earlier, when we sold Adenosine Therapeutics, people end up going out to different companies. Most of them have not left. I mean, if I go out, a lot of those people are still there. And I'll say, in fact, when I got this going, I have all but one of my top people have worked with me at one of the previous companies. So it kind of pulled the band back together in a lot of ways. And particular, some of those people from Adenosine Therapeutics have come back here. So that's a really strength of it. I'd also say in the adenosine space, UVA is actually the land of adenosine. It's funny, Joel, again, that guy who I said was the founder of Adenosine Therapeutics, he took over the program from Dr. Robert Burns when he retired. So Bob Byrne invented a drug called adenosine back in the 80s. It's to this date UVA's biggest money-making patent ever. And so if you look around the world at people that have worked in adenosine, most of them have a tie back to UVA. And at one point, when Joel took over, he had that program, almost $200 million in grants were running around adenosine work. And so being here and focused on adenosine makes a lot of sense. And I'd say we also, with the universities around, we have 2 collaborations going on, 2 grants with Virginia Tech. We're looking at working with VCU, have a number of exploratory work going on there for some of the future and pipeline products. And so there is at least enough of a critical mass of the personnel and technologies to do here. I would say you don't have the institutional investors like the venture capitalists. I mean, they just aren't there, though there are a few that are kind of starting to invest in. Paul Manning, first of all, he's a billionaire that has done a number of things, including life science companies. He just opened the Manning Institute at the university, in Charlottesville, affiliated with the University of Virginia. And so there's that brings a lot of kind of funding credibility, and a lot of his team now are doing things in the area. So that's changing. And then the last thing that I have seen absolutely happen since COVID is it used to be that you needed to be within, they literally told us, you need to be within an hour and a half drive of the venture capitalists if you expect them to really be engaged with you and have potential. Back in 90s, we'll say a long time ago when I was graduating from business school, that was kind of a sort of a rule of thumb. And since COVID, that's just not the case anymore. The venture capitalist only, the financier only needs to come in person maybe once, twice a year, and they feel like they can really keep their finger on what's going on because of Zoom and the use of video conferencing. Mm-hmm. So, and a lot of the financiers started, during COVID, started moving away. They're like, I don't need to be in New York. I don't need to be in Boston. I can go to a lot of them into Florida. And so they have changed their lifestyle also. So I think a little of that has changed. And even if you think about what's, going on with Venture South and how the network there is building, you can do a lot without being exactly in the same location. And I see that happening. So we'll see how far that gets, but it's definitely changed.
Yeah, great. And do you think that's also true of the licensing and exit side of the equation? Do you still need to be within a driving distance of big pharma companies to have those conversations, or are they also receptive to Zoom and long distance collaborations?
Yeah, I would say it's even more so the case that has changed with pharma. Investing can get very personal. And so, literally getting in a room with somebody, I think I still, in my personal opinion, it's still important. I think most of the venture capitalists believe that. They just don't need to do it quite as often. The pharma companies have, you often don't even have to meet with them in person. In the old days, you had to go to New York to meet with Pfizer. You had to go to various places. And I'd say today, that's not the case at all.
Okay, good.
Yeah.
So not at a disadvantage later on when you get to that point of needing to or wanting to have those relationships.
Yeah, and they're fairly, the pharma, the acquirers are fairly spread out a little. The finance, the big life science venture capitalist firms are still fairly concentrated, at least their headquarters. The pharma companies have really moved. They've literally moved around their headquarters and such, and they're not as concentrated as they used to be.
Okay, got it. It's a topical subject around non-dilutive funding and the challenges of accessing that and the reliability of that and the processes around that. Is that impacting either your core drug focus or any of the other pieces of the portfolio under development?
Yeah, so on the core drug, I took a strategy. We're applying for some grants, but nothing we do is dependent on grants for the core drug. For the other programs, the actual strategy is use grant money to fund those. We've won 6 grants, $5 million in the 2 years since we got going. So we've been relatively successful. And that comes from having Dr. Joel Linden, Dr. Robert Thompson. We find luminaries in their fields to be our collaborator. When I say we use academics, we really get big names, and you put the 3 of them on a grant, you have a leg up. I will say, though, that grants have definitely gotten harder over the past 25 years. And in particular, I'll mention the SBIR grants have gotten way harder. And I actually feel like I have a pretty good feel for what's gone on there, is that academics realized that the SBIR had higher pay lines, meaning it was easier to win the grants. And so they have created companies, and not that there's anything, I mean, academics have total capability to run companies and all that. So it's not about that. It's just that the competition coming from academia for the small business loans has increased dramatically. Also, because of that, the composition of the review boards on the SBR programs now is significantly higher. Those that are dual hatting business and academia, where it used to be they were primary, there's just a lot of business people on there, and you have more big pharma and all that. You just don't see that many big pharma on the SBR review boards. And also for the small business, that's a piece that's really, I don't even see as much of an advantage over an R1, which is the standard academic grant. Getting into some of the invitation grants where you can show a whole bunch of data along the way, and then get the interest for, and it takes a long time to get the interest from the NIH or researcher there that says, okay, I've been following you, this looks good. I'd like to see if you can support. We're working on some of those, and we'll see if we can be successful there.
Okay, got it. And so I think I missed it when you first mentioned it, but how many things are there in that wider portfolio? How many near-term candidates or dream candidates are there in that portfolio?
Well, so we have, I would say, in that portfolio, two things. There's a couple of things that we're working on to potentially use our lead product. By the way, that product's called ADO5030. If we have any, and this is both as a backup plan and a value enhancer, and then finally to do some human testing so that when we bring up our next molecule, it would give us a leg up. So we're looking, how we might also use ADO5030. So that's kind of one part of the pipeline. And then the second piece are other molecules. So it'd be another 2B antagonist, an A2A agonist, which would be anti-inflammatory potentially for autoimmune disorders, and then A2A antagonist, which is, so that those other two are on the 2A. Remember the A1, the 2A, the 2B, the 2-3. We're going to go after first the 2B antagonists, and then 2A agonists and antagonists. And so we have about 10 things that we have 10 different academic collaborations going on already, that we are trying to generate data and figure out, what our next one will be. And I would say at the moment, I really like pain, migraine, and lupus as potential next targets.
It sounds like you can solve just about everything with this platform. So that's pretty impressive. Okay, great. Well, so just maybe a somewhat negative question to end things on. What do you think the biggest challenges are for you now, having raised the money and having got the last doses in? What's the next biggest challenge you're facing over the next 6 months or so?
Yeah, well, first of all, we believed this. We were confident this would be safe, but confidence is not the same as certain. And so I would say at the moment, I'd like to just take my breath and say, we got safety today. We're really happy, but coming forward, we need to do this human proof of concept study in asthma. Based on the theophylline data, based on a number of things we've done, we feel like the risk is low. But until I have the data, that's the big one. If we are successful there, then I think the world is our oyster. If we are unsuccessful there, then we got to grind down and develop one of these backup programs. And so at the moment, that's my challenge is get human proof of concept in another program with our lead molecule, while at the same time, or be ready to at the same time as we're developing the asthma drug. So I like to, my belief is management, one of your main jobs is to create options. I frequently say there's no such thing as a bad option. There's only options you don't take, right? So we want to be creating options so that at any point in time, we're not dependent on any one thing.
Okay, got it. And is there anything that listeners or Venture South members or anybody else can do to assist you at this point?
Well, I will say, with what I just talked about there, with the opportunities for a number of things, we are funded now for doing our asthma program. We're funded to go into the future as we need. With success, and by the way, we've already have, I'll just say, banks interested in representing us to take us out. So I'm planning on running a process that we would launch hopefully in the spring to sell the company. And that'd be, one of those things where you try to create an auction situation. But now that we've wrapped up this round, we're starting the work to do a next round, and that the funds from that would be to explore some of those other opportunities. So keep your wallet, near if you have interest in looking at that in the future, and we think we have a lot of opportunities here. Just also maybe the last thing, we're targeting things that are major diseases. And if you look at the last 18 months, there's been 5 asthma deals for phase 1, phase 2 products. And the lowest valued deal was $850 million. And so we believe that there's actually a real potential for significant upside with the first product, and then with each product after that. So we're not thinking small when we're going here, and we'll be in touch. And I'll be calling Venture South one of the first ones as we get going.
Great, we'll be happy to take the call, and we'll try to find some other people with some dry powder to keep this pipeline moving along speedily. Well, Bill, thank you. I really appreciate your time today. Thanks for all the work you're doing and for spending a few minutes with us, and best of luck on the next few months at Adovate.
Yeah, Paul, thank you.
Thank you for listening. Visit us at ventureinthesouth.com for a complete list of previous and future shows.